A blessed day to everyone. My name is Jose Flores, and today we are going to share with you the protein structure that establishes the basis for its interaction with other molecules in the body and hence defines its function. This video will go through the different protein structures. The first topic on the list is the primary structure that will be discussed by Shawn. The secondary structure will be discussed by May. The tertiary structure will be discussed by Trisha. And the quaternary structure will be discussed by Dorcas.
The simplest structure of a protein is a primary structure. It is composed of amino acids which are linked and held together by peptide bonds. This type of structure has many amino acids, therefore, it is only right to call the peptide bonds polypeptide. DNA genes in a protein determine the order of the amino acids. The gene of a person plays a crucial role in how the structure of the amino acid plays out. If a person has an inherited disease, then the structure of the protein will change significantly. An example of this is a sickle cell anemia disease. This disease involves the change in a single amino acid in a hemoglobin chain. A normal hemoglobin chain consists of about 600 amino acids. The single change that the sickle cell undergoes is a substitution of valine with glutamic acid. One change in the structure may not seem very significant, but that one amino acid completely changed the structure of the protein. This caused the blood cell to become a sickle cell-like figure. A person with sickle cell anemia would have a hard time distributing blood throughout their body because of the fact that the sickle cell blood cells would get stuck and clogged up in the blood vessels. The sickle cells also die early, causing the body to have insufficient blood.
Secondary structure. The secondary structure of proteins is determined by the pattern of the hydrogen bonding. A large number of servers and tools are used to predict the secondary structure analysis. The secondary structures play important roles in protein structure and protein folding. The secondary protein structures depend on the local interaction between parts of the protein chain, which can affect the folding and three-dimensional shape of the protein. There are two main things that can alter the secondary structure. Alpha helix, a three-dimensional spatial arrangement of amino acid in a polypeptide chain, held by H bonds between the H and H group of the oxygen atom of a carbon atom double bonded to an oxygen atom of the fourth amino acid down the chain. A corkscrew shape that looks like a coiled telephone cord. Beta sheet consists of polypeptide chains arranged side by side. Has hydrogen bonds between chains. Has R groups above and below the sheet. It's typical of fibrous proteins such as silk.
The tertiary structure of a protein refers to the overall three-dimensional arrangement of its polypeptide chain in space. It is primarily due to interactions between the R groups of the amino acids that make up the protein. It is generally stabilized by outside polar hydrophilic hydrogen and ionic bond interactions and internal hydrophobic interactions between nonpolar amino acid side chains. It involves covalent bonds and dipole-dipole interactions. Also important to tertiary structure are hydrophobic interactions, in which amino acids with nonpolar hydrophobic R groups cluster together on the inside of the protein, leaving hydrophilic amino acids on the outside to interact with surrounding water molecules. Tertiary structure is the folding together of secondary structural elements and other unstructured protein strands, creating a globular protein with a particular shape. And finally, there's one special type of covalent bond that can contribute to tertiary structure, which is the disulfide bond. Disulfide bonds, covalent linkages between the sulfur-containing side chains of cysteines, are much stronger than the other type of bonds that contribute to tertiary structure. They act like molecular safety pins, keeping parts of the polypeptide firmly attached to one another.
Several proteins are composed of a single polypeptide chain with only 3 levels of structure. Some proteins, on the other hand, are composed of numerous polypeptide chains commonly known as subunits. These subunits might be homodimer or heterodimer. When these subunits join together, they form the quaternary structure of the proteins. The quaternary structure describes how these protein subunits interact and arrange themselves to form a bigger aggregate protein complex. The protein complex ultimate form is once again maintained by interactions such as hydrogen bonds, disulfide bridges, and salt bridges. Proteins fold into certain shapes based on the sequence of amino acid in the polymer, and the resulting 3D structure is directly connected to the protein function. Hemoglobin is an example of quaternary structure since it carries oxygen in the blood and is made up of 4 subunits, 2 each of the alpha and the beta types. The same kind of interaction that contribute to tertiary structure hold the subunits together in quaternary structure.